title is In Vitro Inhibition of Babesia bovis by a sulfone cysteine protease inhibitor. To start off, I will talk about a little bit about Babesia and babesiosis. And Babesia causes babesiosis in both humans and animals, and these infections can be fatal. And Babesia is a tick-borne and blood-borne parasitic disease, and the species that causes human babesiosis is called Babesia microti, and this is prevalent in the northeastern United States. And it's becoming a serious problem in blood transfusions because there's no way to screen for the parasite in blood. And these transfusions are given to neonates and individuals who are sick and maybe immunocompromised, and the disease it causes, it's really serious. And this is a picture of a stained blood smear of blood cells infected with the parasite, and it's just what it looks like under a microscope. And babesiosis can be life-threatening, and it's a malaria-like illness, and symptoms, it causes flu-like symptoms, which include fever, chills, headache, nausea, and fatigue. And complications that occur include severe anemia and low platelet counts and multiple organ failure. And the disease is potentially fatal. And it's important to, there's no drug that's available that's specifically used to treat human babesiosis, and currently antibiotics are used to treat these infections, and it's usually a combination of different antibiotics, and this can cause the patient to become really sick because antibiotics are meant for bacterial infections, and we don't know exactly how the antibiotics work to kill the parasite. And also currently to treat bovine babesiosis, imidacloprid is used, and this drug it works fairly well, but it's not approved for human use. And so part of my research my project, I used Babesia bovis as a model system to evaluate the efficacy of a drug, a cysteine protease inhibitor called K1177, to see if it inhibits parasite growth in cell culture. And so because there is no treatment, no effective treatment to treat human babesiosis, this is really important to find a drug that we know how it exactly works and to treat that's specific for treating the illness. So K1177 is a sulfone. It's called this because of its chemical structure, which is shown here. This is what it looks like, the chemical structure. And the drug targets papain-like cysteine proteases. It's a family of proteins that are important for a number of different functions, such as parasite growth and survival, and host cell invasion, so during infection. And in Babesia, there are two cysteine proteases, and they're called bovine 1 and bovine 2. And this drug, the drug targets these proteases and kills the parasite. This drug it's been shown to be non-toxic in animals and humans, and it's already undergoing phase 2 clinical trials for the treatment of Chagas'disease, and which is predominantly affects South America, and it's really serious because it causes heart disease and cardiac damage in humans, and it affects a lot of people. So research on a number of parasites that cause malaria, Chagas'disease, and sleeping sickness, and Schistosomiasis, which affects hundreds of millions of people worldwide, has shown that this drug works on these parasites. And it's already been shown to cure Chagas'disease in immunodeficient mice and protects dogs from cardiac damage in Chagas'disease, and it's been tested on malaria and the parasite that causes sleeping sickness and Schistosomiasis. So this is the structure of the two B.bovis papain-like cysteine proteases, and these diagrams show what the active site of these proteins look like, and as you can see, they're very similar. And this domain is conserved among the parasitic organisms that cause malaria and sleeping sickness and Chagas'disease and all these other diseases. They're conserved, so that's why the drug works against these parasites. And these proteins are really important for a number of different functions and for parasite growth and survival. And in my experiments to test the drug, I tested the drug at a number of different concentrations over a period of 72 hours. And each condition, so I tested the drug at 1, 10, 15, 25, and 35 micromolar. So they're just different concentrations of the drugs. And I tested the drugs on B.bovis infected red blood cells. And I would collect samples at each time point, so at 0, 24, 48, and 72 hours, and use flow cytometry to quantitate the samples to determine the amount of uninfected and infected blood cells in each sample. And this is done by adding a dye to the samples, preparing it and adding a dye. So I would have, the red blood cells don't have metabolic activity, and Babesia bovis metabolizes the dye, so Babesia breaks down the dye into a product that fluoresces. So you have infected cells with Babesia that fluoresce, and uninfected cells that don't fluoresce because red blood cells don't have metabolic activity. So you put this into a machine, and it uses a laser and a computer to determine the the proportion of infected and uninfected blood cells based on fluorescence. So infected cells fluoresce, and this is the graph that you get from this technique. And this here is a graph of the drug at 50 micromolar at 72 hours, and on the left side of the graph you can see that the left side of the graph represents the proportion of cells that are uninfected, and there's a lot more, it's at 99.72%, and this side shows the amount of cells that are infected, and it's at right now it shows it's at parasitemia at 0.28%, which is very low. So it shows that the drug at 50 micromolar is killing the parasite. And from this flow cytometry graphs, this data, I used it to make a graph, and this is what it looks like. So here, appropriate controls were used in the experiment. So this line here represents the no treatment control. It's the negative control because I don't use any drugs or anything. I just treat the cells with media, and it doesn't kill the cells because there's no drugs or anything in it. And then the orange here, it's the middle carb control. It's a positive control. It's actually under this pink line here. So this drug, we know it kills the parasite. So that's why this is the amount of the level of parasitemia. So the level of infected cells. The drug kills the parasite, so it's all the way down here. It's almost at 0. And these lines represent the different concentrations of K1177 that I used in my experiment. And this line here is the 15 micromolar, this green line here, and then this is 10, and this is 25. And as you can see, the drug works really well at 35 micromolar, which is the purple line, and 50 micromolar. It's all the way down here. So there aren't a lot of infected blood cells at 72 hours when the cells are treated at these concentrations. And yeah, there's no significant effect at 25, 15, and 10 micromolar. So, yeah, the drug works really well at 35 and 50. And the next steps to be taken in this research is to test the drug on a human Babesia species in vivo. So, human babesiosis that affects the northeastern United States is caused by B. microti, and we would use this in a mouse model to test the drug in a mouse model, and also identify the IC50 and make sure it's not toxic for the host or the animal being treated. And the IC50 just refers to the concentration of drugs that kills at 50%, kills 50% of the parasite. So, and in vivo. And we would also confirm that B. microti, which causes human babesiosis, has the same drug target. So this is the papain-like cysteine proteases. And to do this, we would clone and sequence the parasite, the genome of the parasite, and compare it to the genome of B.bovis, which is already complete. And also the genome of B. microti is really small. It's much smaller than Babesia. That's why we have to do this, to make sure it actually even has the protease that we're looking for, which the drug targets. And we'll also need to determine the specificity of the drug for the cysteine protease target. And this could be done by expressing the protein in a different models, animal, a system, and to see if the drug binds the protein in like an inhibition assay. And I would like to thank McNair and the program and staff, and Monica Padroni, who's the lab manager, and she showed me how to use flow cytometry and like how to do all these experiments, and she taught me a lot. And my mentor for all her help and guidance and with this project, and also our collaborator, Dr. Kirkwood Land at the University of Pacific in California. And this project wouldn't have been possible by the funding provided by the Wellcome Trust and the United States Department of Agriculture. And are there any questions? Thank you.
Tracy, would you expect any resistance from this type of drug, and like at what concentration would you expect that at?
Well, this drug, because my experiments show that it doesn't work, it only works at really high concentration, so it might not be, well, I don't think there will be any, because it's really specific, and it will be good.
Is there any side effects of using it like such high concentrations? Is there a difference in like side effects at all?
Well, it's already undergoing phase 2 clinical trials, so it's already, all that's been done, and that's like a late stage clinical trials, and it's already being used in humans, so it's not toxic to humans.
What was the most difficult part for you?
Just, I don't think there was. It was pretty straightforward. The process, the experiments were really straightforward. So, yeah.
Any other questions for Tracy? All right, Tracy. Very good. Very good. So, as is a tradition, we'll go ahead and give Tracy. Where do you want Tracy? Her McNair pin. Congratulations.